a couple announcements. If you guys weren't here last week, we had the Innovative Contest. Mitchell Schumann was the winner. Mitchell Schumann's actually the only entrant, so he was winner by default. Regardless, he still had an excellent proposal. It was about technology that would unite readers or academics together to collaborate on projects across universities and across the country. I don't think is there any announcements for PBSGO, the organization, the Plant Breeder? You had the meeting Thursday, right? Okay. So then today's speaker is, it's on synthetic hexaploid wheat, and it's by Dr. Okay, I will let you do the introduction.
Okay. Thank you so much, Lauren. Well, it is my pleasure to introduce Dr. Mujib Qazi. And when you think about CIMID, the National Center for Wheat and Maize Improvement, the Green Revolution, Dr. Bollock, you also think about Dr. Rajaram and you think about Dr. Mujib Qazi, because Dr. Qazi was the director of the Wide Hybrid Program, the Wide Crossing Program. And he is the father of synthetic wheat. And synthetic wheat actually now are in more than 75% of CIMID germplasm. And CIMID germplasm is grown in like more than 55 countries globally on over 60 million acres of land. So a lot of that credit is to be given to Dr. Qazi. And I'm not going to steal his thunder. He's going to talk about that journey, which is unfinished. And by the way, Dr. Qazi yesterday signed the acceptance of that junk faculty status that we gave him. The faculty of this department unanimously voted to bestow him with the adjunct faculty status, and he's going to be active with us on continuing this journey with synthetic wheat. Dr. Qazi, we are honored to have you here today. And by the way, this is working now. Yeah. You have it, so you can.
Where did I keep it? It's gone from here.
It's over there.
Oh, it's there. Yeah, okay. Good afternoon, everyone. Dr. Ahmed, thank you very much, and the faculty here for inviting me over. And it's a pleasure to be back in the United States and give the seminar. The current trends on the global end have made a lot of projections. Looking at 2025, 2030, across zero hunger, sustainable development goals, the 2050 vision of food security, the population enhancement then to 9.2 billion. Another 5 years after that, we touched 10 billion. And across the board, there have been many advocates who have suggested that we should now somehow try to access the global enriched alleles of genetic diversity. I'm going to take you over a journey over time on which I started this effort with my colleague at Kansas State in 1964 when I was a landed immigrant and I went to Kansas State. It's 2015 and I think still it's an unfinished story. That particular quote of George Bernard Shaw really excited me. I was reading the memoirs of John Kennedy, and at the caption of one of the chapters was this quoted, and it applies to white crosses because I like to do things which are absolutely considered impossible. White crosses had a very checkered history earlier on. Over the past 100 years, 150 years, not many hybrids were around. Easy combinations were made by Dr. Sandu in Oklahoma State, and those of the agropyron intermedium and elongatum, but the complex ones were not around. So that's what triggered me to try this venture. White crosses got started at Kansas State through Lynn Bates. His classic paper on proline estimation from Purdue when he was a master student, Mertz, Bates, and Young, one of the most highly cited papers in the Science Citation Index. He went on to Mexico and he got some ideas, interacted with Dr. Bollock when he was there setting up the protein lab in Chapingo, and came back, pursued his PhD, and he was a student when I was a postdoc there. When I came back in 74, he had become an assistant professor, and he shared with me what he was doing. He said, I'm crossing barley with wheat, wheat with oats, barley with rye. And I just immediately blurted out, I did not have the class, I guess. I said, well, that is absolute hypocrisy. I don't think you can do these combinations. I said, how are you doing it? And he said, well, I'm using immunosuppressants, and they eliminate the incompatibility barriers. And I said, Len, I'm going to prove you wrong, because what you got are chemicals you put on, and they are serving as chemical mutagens, and that's what you are saying. And he said, okay, I offer you a small stipend, you stay on, prove me wrong. So I used barley as a test system. I tried for about 4 months, get the crop, grow out the M1s, see the chlorophyll mutation frequency. I'm pretty sure you'll see the chlorophyll mutation and I'm on my way to find something else. Well, I failed at that. And then finally came the big Rockefeller Foundation funding. On Christmas, he gave me an envelope with a check on it, and he said, well, here's a contract and your check ahead, and you're recruited on. So I started White Crosses. We had limited success, and I'll share that in a few minutes. CIMMYT got directly involved in White Crosses. They were doing it here and there with Lynn in Kansas, up to 1979 when I moved to Mexico upon the invitation of Dr. Borlaug. The mandate given to me was pretty simple. I expected a long lecture from Dr. Borlaug and Dr. Anderson, both late now, and they just said, well, CIMMYT has got a bad name on White Crosses, and people are saying that Lynn's approach was nothing but similar to Lysenkoism. So you get us back the scientific credibility in this area. We give you 2 years to find your feet and see if you can make a suggestion, do we carry on or we don't. And he said, if you are successful and you give us the plus on it, then you've got 2 things to consider. Number 1, do creative research. I mean, his classic words, do not become an ivory tower scientist or build castles in the air or chase academic butterflies. And second, make sure that you are not going to do anything which the classic universities are doing in the United States. You have to be applied. Remember the farmer in the field. The measure of your research is going to be tons per hectare in a farmer's field. You don't do that, you really done nothing. That's typical Norman Borlaug. The historical background in terms of review of literature that we have was very, very scarce. The classic 1872 paper by Rimpau on the first fertile triticale. That was the headliner for White Cross. Then Bob Farrar, 1904, in Australia, published on a wheat barley hybrid, and he gave it the name Bob's. And no cytogenetic evidence, and refuted by everyone. Of course, then the historical discovery that the D genome was derived from Aegialopsis scorosa or Triticum tauschii at that time, now called Aegialops tauschii. McFadden and Sears, 2 papers in General Heredity, 1944-45 A and B, and that proved that yes, the D genome is the authentic donor based upon chromosome bivalency. And of course, then from Denmark, my buddy Anton Kruse published internal reports 1967-69 and 1 paper in Hereditors on the wheat barley hybrids, and he was crossing wheat with barley, barley rye, wheat oats, etc. Unfortunately, I saw his thesis when I met him in 76, a big about 500-page thesis, and he was failed by Sir Ralph Riley because he could not believe. I think Anton was way ahead of his time. The second part of the historical was Kansas State started off in 1972. Lynn Bates had published a paper in economic botany, and that got White Crosses going all over. I joined him in 1974, and we carried on. CIMMYT at that time was latched on to Kansas State, and I was based in Kansas State from 72 to 79. However, not very happy. In 1979, on Norm's invitation, I went to lead the White Crossing program, and we started without any objectives, just crossing anything with anything to see if it works, and just the cytogenetic evidence, and that was intergeneric hybridization. Later on came on interspecific hybrids. In '79, it took me about 2 years to make the program, give it a global perspective, and I was very comfortable. What triggered me to go into interspecific hybrids was a freak event. I had a seminar in Washington, D.C. It was a USAID planning meeting on biotechnology. And one of the CGIAR senior scientific peers told me, he said, I liked your talk. You had this title, Vision to Year 2000. He said, my dear Kazi, you're not going to last that long. Maize white crosses is going to fold, and you are next on line. The reason being, not the science or anything that you're doing, is because the funding is going to be diverted to biotechnology. I said, what do you suggest? He said, well, you're the scientist, you do it yourself. How you figure out how you handle it is your problem. But I would just say, give them a run of their money, or integrate, or force them to come into you. I said, all right. So that's how I got into interspecifics. At that time then, we had already approached 1982, and the program mission was set, you need to have a direction. I was exploring new genetic diversity, yes. I had the vision at that time that we are going to do through white crosses, put in different genes, different structures, different traits, and then devolve the varieties around gene deployment and varietal deployment. We don't want a situation like in many countries, you have one variety dominating the show. If something breaks down, you've got susceptibility all over. And so that was kept in consideration. The germplasm that we would generate out of a program in CIMMYT should be a source of human resource development and not isolated but global. And we must combat all major stresses, biotic and abiotic, as best as possible. And of course, you have experimental plot yields of 9 tons per hectare. National averages somewhere are 2.5, some 1.9, etc. So why is this yield gap? So we got to eliminate that. So these were the guiding lines. A research structure fell in place, and normally we separate or stagger research structures. Some just do basics, some strategic, and some strictly applied. In that category, you can say the master's and PhD students and postdocs and several places, you just have a specific topic and you work it through, and the ramifications you don't see too much. The applied people just keep on breeding in the release varieties and they ignore the other thing. So I said, well, let's add a different flavor to the program. So we covered basics, strategic, and applied, dovetailed each with each other, and where the weakness was existent in the program that I was leading, I decided to pick partners from around the world who had particular strengths, and we integrated all of them. So the beauty of this was integration of research across the best professionals around the world. 1980s then, I go from 1981 to 1990, into generic hybridization. I think we reached a stage where we could easily say we can cross anything with anything. That was no longer a problem. Interspecific hybridization got started. It was Bikram Gill's group at Kansas State who picked up this last line here, direct crosses, where susceptible wheats were crossed with screened Aegilops tauschii accession. Precise experimentation, precise transfers, very well documented, excellent for molecular work, because all transfers go into the D genome, and with the D genome specific probes you could tag the transfers easy. Bridge crosses was the one we adopted in CIMMYT, so we don't duplicate anything which Bikram is doing. And this came at the insistence of Sanjay Rajaram, my colleague, 40 years together, and his concept was, I don't want the fancy work of just using Aegilops tauschii. I'm a breeder. We are going to drop our bred wheat by durum wheat crossing program, because if you make synthetics, then we would get the durum variability from your synthetics plus the D genome. So that is what the structure was in 1980s. The first 2 decades, this is what we accomplished as a group with international partners. There are many players in this. I don't want to single out anyone, and I don't want to take credit for anything myself. It's a whole bunch of people. A lot of comments and suggestions and criticism which paved the way to progress. Over this time, synthetic hexaploid wheat production was oversimplified. There's no real science left in it. Output and foundation, yes, The outputs that came out of this program has set the foundation for wheat breeding, and we can safely look for many decades ahead. There are tons of synthetic hexaploids floating around. You all are familiar with the D-genome synthetics, and I'll share with you something else. The third very important aspect when you run a research program of the magnitude that you are in CIMMYT, Norm Borlaug and his legacy and everyone else, that you share germplasm. This is getting hard and hard to access these days. There's a price tag on every envelope. You get a packet of 15 seeds and you pay $15. This should not happen, but circumstances, because money is available for everything else except for research, and the poor researchers or program leaders are compelled to distribute seed. Of course, there's a cost for distribution. The other significant contribution in these 2 decades, I would say, is the promises that we have based upon these synthetics. Practically every abiotic and biotic stress, you have the genetic diversity in this germplasm. The only one which I have found missing so far is aluminum tolerance, and chances are that the Aegilops tauschii route, where it originated from and spread, did not have that problem in the soils, so you don't have any Aegilops tauschii accession looked at so far. And I think we looked at the entire core collection of 450 and accessions over 750. The ramifications, those are some publications. You could get my email from Dr. Amir if you're interested, and I could send them. The first one there by Modan Das, we just published in Genetic Resources. That's on synthetic hexaploids for different biotic stresses. And the one right at the end, on the first one, FO, that's Francis Ogmanaya, and we published a chapter with many authors on it, and this is in Plant Breeding Reviews. It's a very exhaustive survey and review, and our work as well on synthetic hexaploids, and it covers the whole spectrum, right, and it's for students particularly helpful. What were the salient outputs? Now I'm gradually getting into some technicalities here and there. The D-genome synthetics, when I left CIMMYT in 2004 October, we had 1,014 D-genome synthetic combinations. And that's the genomic formula. That comes from tauschii. Then we had Gina Gadira at Kansas State who said, well, all these are spring habit or facultative. Could you make us some which have a winter habit so the winter wheat breeders could get into it? And she sent me the winter habit durums, and we made her some 84 of the D-genome synthetics of the winter habit. Moonlight madness, I should say, in order to get funds, the funding from CIMMYT was coming to my program through the D-genome work which was highlighted. And had to play it smart with the directors and the trustees. Said, all right, get the money and use it somewhere else and get some results, and then you move on. But the job should be about 80% done before you solicit the money. So that gave us the 194 A-genome synthetics. When this was finished, I got the money for this, and then I went into the B-genome synthetics. Now you see this combination. The A and B's come from the durum, and these two A's come from the A-genome diploid, that's Triticum, Monococcum, Urartu, and Boeticum. The B-genome, Aegilops peltoides, is talked about a lot. It's from the cytopsis section. Cytogenetically, it's a huge problem. It disturbs the chromosome pairing and fertility. And so you got AA and two B's and S coming from peltoides. When you see this, you will say, well, you got 4 A genomes. The cytogenetics is going to be bizarre, and the same way here. And apart from that, this is peltoides. So how do you expect to get fertility, and how are you going to breed with it? That is the textbook knowledge. Fine, we accept the textbook knowledge, that's the foundation. But you have to be creative and do something different. So it was a gamble. Our initial focus, however, was on this, and this was very highly publicized.
Dr. Kazi, I have a quick question here, just to make it clear for those who are not familiar with the wheat genome. Can you just shed light on the original wheat cross that happened in nature, like 10,000, 12,000 years ago in Turkey and Syria?
Good.
What were the 3 genomes, and the fact that synthetic wheat means that you're recreating this cross?
Okay. When I said a little while ago, production of synthetics is, there's really no science in it. We take lessons from history. The origin of wheat is based upon an event about 8000 BC. A durum, a bee genome diploid, it's a grass with the bee genome, was accidentally mated with the A genome pollen donor. You got the hybrid, which was BA, no colchicine at that time. It doubled spontaneously, and you had the fertile product BBAA. This then in 6000 BC more or less was hybridized onto the durum, the two Bs and two As, and you got a F1 hybrid BAD, BAD. Again spontaneously doubled, and that became two Bs, two As and two Ds, and that was the origin of wheat. How many events happened? There's a paper by Tolbert that this original crossing, 6000 BC, carbon dating experiments and whatnot, there were two events which happened. This is very important. My advent into interspecific hybridization, I might as well say that now, and I delivered that seminar in Washington, D.C., had the whole morning free. A bookstore, The Origin of Species, a book had come out. $160, I bought it. Got on the plane to Mexico, started reading. Second page, left-hand side, was a schematic of origin of wheat. And the fine print said, Tolbert et al., two events of Aegilops tauschii. And literature work earlier on told me, if you got 750 Aegilops tauschii accessions spread from Turkey all the way down into Afghanistan, that route, then they must have tremendous DNA polymorphisms, etc. So this was in mid-80s. So I said, all right, that's the easiest way to link up with biotechnology. So that's the start. Now let's go back to these 4 A's and 4 B's. The production process, you can keep the durum as the female, with tauschii is the pollen, hybrid, double, and Aegilops tauschii is the donor. You could reverse this cross also, tauschii is the female. It's advantageous. If you are an embryo culture lab and you're a wizard at it, play around with the media, this crossing gives you somewhere around 10 to 15% seed set or embryo accession rate. If you reverse the cross, the embryos are very ill-defined and you get about 85% success. We didn't have the time to waste on doing that academic thing. We carried on this way. Here is the second one, durum, A genome, hybrid, doubled. And these are the 3 donors. They're all diploids. And here the speltoides, hybrid, doubled, and thus the speltoides. Now those 4 A's and 4 B's, lo and behold, this is textbook knowledge, completely defied. You don't see 4 chromosome associations. These would add up to 21, and this is called a chromosome pair. It's called a bivalent. So you got perfect 21 bivalents, and that said, well, it's simple. Now you got regular meiosis, and this should have very high fertility, which exactly it does. So the 194 is now at this time the unfinished story, open for breeders to exploit the A genome diversity. And the same thing on the B genome. Because of speltoides, you see some complication. You see in the center a 4 chromosome association. The fertility here is a bit reduced, the seed is shriveled, the endosperm is shrunken, but still it works. You get some fertility seed set, so it's a good stock. It's excellent for breeding because you don't want to get stuck inside your genetics. When we were doing this exercise of synthetic wheat production, the observations were that the durums had a specific role. We were using the durum parents coming from the durum wheat program of CEMET, and we found that it had a crossability factor. Some were better than the other. I've already talked about crossing direction, male or female. Embryo culture, yes, you have to be very good if you the other combination. And then this source and durum, these integrative partnerships are very, very important across multidisciplinary avenues, because then you can open up and you share the germplasm and you can capture all the talent that is around. We made up a targeted route also. Apart from bread, we started looking at durums at the same time. That was moonlight madness research. It was just a fun exercise. And in this, I put in digression for durums. You can improve durums by straight white crosses, but this was something different which brought in the biotechnologists into it. And we did that with taking the durum genetic stock, which has the PH1C gene on chromosome 1 on 5B, stock produced by Benito Giorgi in Italy, acquired that from him. This enhances chromosome pairing, and then we use the Agelop-Stauchii accession with the desired trait in it, and got the hybrid ABD, and this gene is recessive because it is on 5B, long arm. You look at the meiosis, normally if the genomes are separated you will get 21 single chromosomes, but that was not the case. We saw in abundance of cells 7 single chromosomes and 7 chromosomes paired. Then we did the in situ hybridization work on it. Noor Faridi here is an absolute maestro on it. And you get 7 pairs, and these pairs are between the A and the D genome. Now that has the applied connotation of how you are going to improve durum with genes which are never accessible when you cross durum with durum or anything else. And this is just to show you that yes, here these are the pairs, this is the trivalent 3 association, so you got that. So you don't see 14 single chromosomes like this, you see a lot of pairing. And that's the breakthrough we needed. Completely unexplored at this time, and that is one of my unfinished stories. I think the young professionals of today and the future should take this on a good thesis venture and somewhere try to produce this. It will be a very significant contribution to the genetic resources. What is the potential of synthetics? Of course, enhances diversity. This is just wrapping up on that section. Stress coverages across both. Knowledge generation led to publications, etc. Then you have the human resource development. And this is now, I'm looking at it. My daughter is hopefully in the audience. She's actively involved in sustainable development goals and zero hunger programs. And she's a visionist with the group that she is in. And I think in order to meet this, you need implementation processes, and I think this germplasm will fit into it. And for these 2 projections, you've got lots of facets to consider, and this germplasm is very suited to address that. In order to address it, what do you need? Now let us say, all right, we've presented the terms that this is what needs to be done. How do you do it? That's the big question. Pre-breeding is the way to handle it. You got lots of breeders. You have less pre-breeding facilities around. There's a consortium out of England. Ian King and his wife are running it. And we can debate that if you have any questions onto that. There is always a downside. People should be very careful of the past lessons from CIMMYT, and I should say to state also what Bikram Gillis, his group has done. You must recognize there is a time essence. You just cannot leave research open-ended. It has to have a closure, and you must have a product. It must address weeds across all mega environments, and you cannot breed or you cannot put a single trait at a time. You must have multiple stress agglomeration. And of course, that is absolutely essential. You must have applied research objective. I mentioned earlier germplasm sharing and how critical that is. It's based around generating integration between professionals. I freely shared the synthetics in mid-90s. Mark Sorrells came down to CIMMYT. He wanted a few things. He's out of Cornell, as you all know. Gave him synthetics. There was a Mexican student he took on as a PhD, Enrique Otrique, and they developed single seed descent population, Opata by that synthetic, 152 individuals with 2 controls in it. This later went on, increased, and it became the International Triticide Mapping Initiative, which is now handled and managed in California Davis. The first trait mapping that was done on this population was done by Enrique on Karnal Bunt. The ITME was shared with Marion Roeder in Gottesgabe, Germany, and she published the first microsatellite map of wheat in genetics in 1998, and it led to global partnerships and all got together in Victoria, Australia, and that was in 2008. Australians got on the bandwagon and the Chinese too. Now recently you have the DART microarray developed from those synthetic populations, and you had the 9K SNP array and the 90K. And China, just last week, one of my postdocs is on a postdoc tenure now in Beijing Genomics Institute, Uwais Rashid, working with Dr. He. And that is one of the centers which has produced about 90% of the functional markers, SSR markers. And they have come up with a 660K chip. These synthetics have also been advocated by many people. They are saying that we are talking about a yield plateau. Where do we go from there? 9 tons per hectare. Can CIMMYT get to 10? They're trying awfully hard. It hasn't reached there yet. It's not consistent. Accidental hits here and there have happened. But you need a breakthrough there, and that's very complicated. So where do you go? And this advocacy of people to look at the genetic resources, access them. I just go one step ahead. It's not a question of accessing them. They already accessed, the stocks are there. You should be wise enough to know how to use them and be very selective, recognizing the factors of time and output, etc., and what it does. So they have been saying that this is the source of the next green revolution. And Dr. Swaminathan, one of my mentors from India, you all know him, he coined a different word. He said it in the same meeting in the World Food Prize once, and listened to the lecture. He said, well, we are not really looking for the next green revolution, are we? We are looking for an evergreen revolution. And I was very motivated, and I had to put up Dr. Swami's name on it. So that's what we are looking at. How do we handle synthetics? There should be some sort of a direction. We've got many young professionals in here and students in here. You must have a beautiful locked-in breeding strategy involved. It could be F1 top crossing, it could be limited back crossing, whichever way, whichever thing delivers earlier. We have not capitalized on double haploid breeding. Do we make the double haploids as F1? If you got lots of money, well, fine, do it on F1s. But it's better to have a round of selections, F1, F2, go up to F3, and then make the double haploids on the F3 population on selected plants from the field. Cuts down the cycle. Instead of rushing up to F10, F12, etc., you can finish it much earlier. You get the homozygosity restored. Bridge crossing is favored because it brings in all the diversity from the durums at the same time. Ideal for wheat breeding and for developing countries to handle that. There is one problem, however. To achieve durability and sustainability, you need, how do you get durable resistance? You must play around with minor genes. And these wild species predominantly have dominant genes, so they could break down. This thing is a very dynamic process, and you've got to worry about that. So there is a solution, and we are going to show that as one of the future area unfinished work so far, and I'll share that with you right in the end, so those young professionals pursuing a degree can take this lesson home, I think. These are the other guidelines which also motivate me even at this time. The 2050 vision of food security. It's a buzzword, yes. You relate this with population, 9.2 billion, then 10 billion. You got so many empty bellies to fill, and all those mandates are laid down. There are 2 programs there, Zero Hunger and SDG for 2030. The first one is for 2050. How do you tackle them? Then there is the changes in molecular science. You've got genotypic platforms coming up, very sophisticated. What is the advantage of most of the countries? And this might not go down with many people from developing countries, but I have been very subtly mentioning that what is your competitive advantage? Do you set up a high quality molecular lab in your countries across the country? With this changing technology coming in, you have laps of water, laps of electricity, and thousands of constraints, and you're going to be full of excuses why you didn't do it. So Capitalize on what your competitive advantage is. And the competitive advantage, from what I have been in Pakistan over the last 10 years, is phenotyping. You got areas of strength of canal bund screening, powdery mildew, you got yellow rust, etc., heat, drought, salinity, sodicity. I think that's what, then you are compelled to link up with advanced labs, partner them on a program and do something together. Never forget that no matter what we do, this is Norman Borlaug, one of the earliest lectures he gave me. I was showing him the translocation of a chromosome 1B, 1R. The variety from Rajaram was very famous. He walked into the lab, he said, I haven't seen chromosomes. I said, I'll show you one. That's why I called you. And he looked at it, he said, yeah, what do I see? I said, do you see the color? He said, yeah, I see one arm of your so-called chromosome of a different color than the others. I said, that's carrying the 1R segment and it's got 4 genes on it. He said, oh, okay. He said, you know, no matter what you do, what is this material? I defined it. He said, good, now you can do it. Because Raja has pushed Viri all around the world, and that was 1982. And he said, you have detected it, and Viri wheats with this particular genetic exchange is all over the world. So remember, the measure of research is this. No matter what you do, that's the measure. Now we are looking at this thing, and we hear a lot about climate change. You will be amazed that getting early wheats has defied the wheat breeders ever since Sonalika came out in late 1960s. To beat that earliness of Sonalika, Rajaram and company and Martin and Ravi Singh and Norm have not been able to do it. Very, very tough. Ravi has just got a line out which says it is equivalent to or a few days early than Sonalika. So that is just one of the factors, earliness, to beat the terminal heat stress. That's very significant. But there are many other factors. We only look at research to solve everything. And there should be a different vision should be in place and emphasized. We all know about it. Management practices and policy setting. The small-scale farmers, their limitations, the land leveling, laser leveling, fertilizer inputs, market prices, etc. So it's a whole arsenal, and that compels you to work as a team and a group right across the different fora that you have. These goals are very well laid out. I mean, these are Ankle Development, Sustainability Development Agenda 2030. Nabiyat, thank you for the information you sent me. Alleviate poverty, alleviate hunger. All less than 3-year-olds should be very well nourished and overcome malnutrition. You're looking at iron, zinc, protein, phytic acid, etc., etc. All that diversity is present in D-genome synthetic hexaploids. It has been worked on by Ivan Monasterio, Robin Graham and Adelaide, and in Turkey as well. So that's going on. But in order to get all this, there are huge challenges, and what I am advocating with many around, I've discussed this with Rajaram, we need implementation of all the good things which these policy set organizations have shown us. They've shown us the path, we've got to follow it, and that's what needs to be done. Let's talk about the future. Can I share with you some direct pointers, so there is some substance to this talk. We need to harvest or harness allelic diversity, and there's a preference there. The D genome, A and B, A is very doable, this is very challenging. In order to put the major genes together, if you've done your molecular work on it and you've identified the diversity in the Taoshi-based synthetics, take one synthetic with diverse from the other synthetic, put the two together, get the F1, get the F2 population, and make new double haploids, and that is called gene pyramiding. So this way you're putting major genes together. They will give you a semblance of minor gene effects. And of course you can use other tetraploids. Many are unexplored. I have another slide later on. And then of course you can use these hexaploids which are not used, like Triticum spelta from Switzerland and other areas. That is homologous genetic exchange. They will fall into chromosome pairing perfect and transfers will be effective. There won't be any penalty effect on yield. Homologous pairing, this is intergeneric. Ian King and his group have been looking at it. They've been crossing it with rye. I just say exploit existing genetic stocks. I haven't written to Ian, but I will. For rye, there are numerous translocations which Friedrich Zeller has got in Germany. I think he should access that and he'll be way ahead. Must recognize that this is 2015. You can't spend time now on basic research. You have to use what you have. This is very clear, partnerships. And this applies to our students here, basic academic outputs and their professors. Research for a student does not end on a thesis and a few publications. It's up to the professor that when that student leaves, the professor must make sure that this takes off and is used practically somewhere. Get another component or stagger the students. So use the arsenal that few students have left on basic research and apply it for the applied goal, as I pick up from Norman Gola. And of course, I've said this earlier, select in your program, that's particularly for the third world, select competitive advantage areas which will allow you to link up with elite organizations. Now to exact specifics, the tetraploids with the A and B genome, virtually unexplored in breeding, very simple to cross, and Triticum polonicum. I have recommended this very strongly for third world countries where you don't have to worry about embryo culture and things like that, controlled environments, all a field operation. If you cannot cross with Aegilops stulcii because it requires embryo culture, you use Aegilops cylindrica. The cross is very simple and you get the D genome transfers that way. No embryo culture required. Hexaploids, Triticum catholicoides and Triticum spelter, excellent for cold tolerance and viruses. People like to use land races. In Bali, it has been excellent. In Ikara, they worked a lot. Wheat land races, I don't think you should be looking here for any sort of resistance for leaf stem or stripe rust. However, if you have some scarcity for a trait like phytic acid and things like this, yes, or high molecular weight subunits, then you by all means do that. You must recognize the faster the transfers between chromosomes of wheat and an alien species, the better you are. More specifics of how do we enhance yield. Translocations, the best example is 1B1R wheats. Several million hectares around the world, thanks to the great work of Rajaram, who crossed winter and spring wheats and got the V lines. And this has gone over several hundred varieties around the world. This is very common to Texas A&M. It originated from Oklahoma, the 1A1R translocation done by Emil Sylvester. Radiation influence in there. There's another translocation for copper efficiency, and this one is excellent for barley yellow dwarf virus and other aspects. It involves the pH 1B locus. This is in a bread wheat in Chinese spring Ernie Sears is doing, and you could use that. These translocations are written up in a chapter I did with my large group in Advances in Agronomy 2013. Again, the students here would like to look that up, and it's extensive paper, about 80 pages or so. Only thing good about it is lots of references, so it adds to your literature. Large spikes buitre. Ricardo in Mexico was studying that, and Dr. Bollock's favorite from early 70s, and now it's 2015. Only thing which has come out is in India by Dr. Singh, a super wheat variety, and one in Chile, which in a winter sort of a habit has given about 18 tons per hectare. Long growing season. It hasn't been adapted to the spring type, but it's very difficult. It's linked with a single culm, as they call it, monoculm, so improvements are very, very challenging. If someone wants to take it as a futile exercise, maybe hit it sometime, maybe, it's good, do it. Stay green character, very common in synthetic hexaploids. 1,000 grain weight of synthetics, 65 grams is very common versus wheat, which is normally bread wheat, 40 to 44 grams. Pentaploid breeding, I go back to the original story why I was disheartened in Kansas State in the first 4 or 5 years. That was failure. Norm was very excited. I know we walked the field in Obregon, and he said, what makes you disheartened with this, Qazi? I said, it's pretty simple. I'm looking at this material and this to me is absolute junk, Norm. He said, why do you say that? I said, I say that because this is not a white cross. Someone in the field pollinated in the morning when the pollen was floating around free, and you had bread wheat fertilized with a durum, you got a pentaploid, and the pentaploid upon further segregation has given you new durums and new bread wheats. That's why you see the phenotypic difference. So there's nothing in it of an alien chromosome. And well, that comment, it may have been blunt, but it paved the way for me to get into CIMMYT. And of course you've got heterosis and double haploids. I wish the breeders would seriously consider to shorten the breeding cycle, get very heavily into double haploid production. Wheat by maize, you get 100% success. Any cultivar, there are maize sources, but you can do it with anyone. It's not, no more science behind it. It's just straight, simple, technical thing, facilities and help. There are private companies who are thriving on double haploid as the breeding, and they do it on F3. Now we see all this array. There's a recent paper in Nature Plants by Graham Moore, 2000, Strategic Pre-Breeding for Wheat Improvement. Look at this chart here. You've got so many, you've got all these SNP markers and platforms, 400 million data points. I sent off an email to China and asked my colleague there and my former postdoc, Uwais Rashid, I said, please make it simple for me. Can we make white cross improvement better with your molecular inputs? And he said, right now, no. It's a desire, but we are not there. We cannot run massive programs to facilitate the breeding cycles simultaneously. That is still tough. But give us time. And he retorted, Qazi, don't forget, you've taken 40 years to reach this point. So at least give me a few. That's just, I think, more late night slide made. You carefully select the diversity. Many countries who are still relying on CIMMYT germplasm, they get the International Bred Wheat Screening Nursery, they take it out. I think that is, in a way, I've watched it many a time, that's injustice. You are not permitting the national breeders to get into quality science and quantitative genetics and breeding and methodologies. They are just taking it, adapting it, this introduction, adaptation, selection, varietal name change, and they say, I released a variety. The credit goes somewhere else, but CIMMYT encourages them, okay, fine, you release it with your name. So they should get more into recombination breeding. The term is come, you produce your own products and then you can barter, you can have good partnerships right around the world. Genotypic and phenotypic selections, excellent, I think you have to worry about it. You must have time-bound targets and you must have accessory factors which contribute to this. And this brings in all the disciplines across our plant sciences into it. This word, I have another slide at the end, I'll show you that. All these relate to basic research. They all have a connection to strategic and applied. This is gene pyramiding between two synthetics of dominant genes, F1 double haploid synthetics, and you get a wide array of them. I listened to some beautiful student presentations this morning. They're using synthetics, they have the pedigree written in, and they have in the bracket after Agelopstauchii a number. That's the accession number. Someone should find the time and the vision to say, if we are using an accession number, let's give it a number 224, and you made the synthetic, and you made the transfers, for you to go back to that accession and find that particular seedling is well nigh impossible. Why? Because no gene bank in the world keeps an accession pure. They keep it as a bulk population. It's okay. It's got a number. So I have been trying. It's a very meticulous kind of a work handling Tauchii. Grow out 10 seedlings, fingerprint them, separate them, raise the seed, and now you've got from each accession 5 seedlings or 10 seedlings, and that is pure stock. And then you redo the program if you can, or you make the direct crossing like Vikram Gill did. Land races. Only if you don't have conventional diversity, Norm and I had a lot of talk on it. It's land races, 1 step forward, 10 step backward. So you bring in a lot of garbage coming in. Like triticum dicockum and dicocoid is a classic example. And that's my last slide here. I appreciate your patience. Thanks to Amir, I was so unprepared for this talk. He gave me a beautiful book last night, and I started reading at 11 o'clock after watching Matlock, went up to 4 o'clock, and I finished the book. I didn't do anything of this anyway. So this is the first time rehearsing on this. Well, this holistic cognizance means that we need to be aware. It's not just straight, simple, a single crop improvement process which is going to fix all these issues. There are many things which we need to broaden our vision on and our strategies. Management, of course, for policy setting. This is very important. So government gets in, you're dealing at a different bureaucratic level, and you've got to handle that somehow. Dr. Borlaug was a master at it. Soil quality, and completely ignored. The microbial flora, the contents in the soil, etc., etc. The micronutrient levels, we ignore them. It's not straight NPK and all these kind of things. We have destroyed our biodiversity. This house, reforestation is an important factor. We have no sense in many countries of harvesting rainwater. You get floods, topsoil flows away, all you've got is plain flat land and your soil is completely lost. Extension services, absolutely miserable many a time. And how can farmers with small holding? In Asian countries, most of the farmers are small holding farmers, about an acre, acre and a half or 2, with no inputs there. So what do they expect? They harvest the seed and they keep the same seed and gradual deterioration and mixing. And that's why the yields are so low. And this one, you should not be isolated into your own research platform. And UG99 is the best example of it. Started in Uganda, moved up, went up to Iran. Pakistan started fearing we've got danger on our doorsteps. India, agriculturists, they were fearing too. It might cross in, go up into Nepal and everything else. Okay, so this gives you the sense to think that national boundaries do not restrict pathogens from advancing. And you have to be very cognizant of what's going on. If Iran releases a 1B1R translocation of what name, Pakistan should not be releasing that. Very careful. And India definitely not. Otherwise you've got a same chain, Kafka's Buho, Kalyan Sona, Bluebird, same pedigree. You're going to be hit all the way. And that's how varieties got hit from Ethiopia right up to Nepal. So we got that lesson to learn. I think I've come to an end. Thank you for your patience.
Thank you, Dr. Kazim. And before we pick the first question from Steph, I just want to mention that because of the joint effort of Dr. Borlaug, Dr. Sanjay Rajaram, and Dr. Mujib Kazi, the semi-wheat occupies more than 68 million hectares globally. And that came through steps. The first one was the Green Revolution. Wheat was very tall. It lodges when you give it a lot of irrigation and nutrients. And the Norine 10 gene that Borlaug deployed, and semi-dwarf wheat, that was the 30th revolution. And then the spring by winter wheat process of Rajaram in the 80s brought the yield to a different level. And then the synthetic wheat that Dr. Kazi produced is in more than 75% of the semi-germ plasm, and it took that yield level further. They were like 20% yield increase over local cultivars in China. We see that also in Texas, and so that was the next level that the synthetic wheat contributed to. With that, we like to pick the first question from Seth.
So, especially in relation to your last slide here, where you're talking about soil erosion and biodiversity, perennials would hold a lot of potential for that. So, have you worked at all on trying to integrate perenniality from many wild species of wheat into?
No, I haven't done that really. I've looked at it. There was a program in Australia which was doing it. Stan Cox, who was at Kansas State, and it's unfortunate he left wheat and he went to grasses.
Sorry.
So he's in Salina, so he's doing that. I haven't stayed with that area, but yes, you could go for perennial, but when we look at the structure around the world, most of the acreage is occupied by regular weeds. They're not into that. It's a whole different area. You could branch off. My sense usually comes from we need a solution and there are hungry people around. But this is excellent what you were suggesting. I think this allows people to get into different areas, and I think they should embark on it. This is visionary research. This white crosses, I mean, even though I'm just an instrument, the concept goes to Dr. Borlaug when he got the Nobel Prize in 1970. In his address, and then when I was counseled by him, he asked the question, he said, are we going to start looking for diversity when we see a problem? Or should we have a vision to see 20, 25, 30 years ahead? And I said, no, why do you say that? He said, well, there are problems which will come up. And sure enough, after he said that, 10 years after that, we had the famous canal bunt problem, which came in, and nurseries from Simmet were blocked into the United States. APHIS got active, and quarantine went all over the world. And we were not prepared for that. So then it took about 6-7 years to cope up with canal bunt.
Okay, we will take the next question from Dr. B.B. Singh.
Dr. Kazi, thank you very much. Fascinating work, and I think you have contributed quite a bit to diversify the wheat, the germplasm. But some of you, as you mentioned earlier, one of your colleagues warned you that come 2000 and biotechnology will take over the white crosses. So that's my question. What's the future of the white crosses versus the biotechnology? And let me give an example. Biotechnology started in 1980, and most of the work you are mentioning also was from 1980. And they go to anywhere in evolution, bring a gene and put anywhere. Whereas your work, our cytogenetics work, is still restricted to gene pool third or whatever it is. So, and students right now are not, many of them are taking white crosses. So what is your projection about our ability to go anywhere in evolution, bring a gene, put wherever we want, versus restrict to white crosses and do whatever we want? I mean, for future training, research programs and so on, I would like to have your comment.
I'm very open-minded as to how you're going to use these genes. If you are alluding to transgenics and transformants, by all means do it. But again, you have many things to watch around the world. The acceptance of that product, I was visiting a program yesterday. Yes, works fine in cotton, works fine in many other things. I did ask the question to that great researcher. I said, why not wheat? He said, well, there's a whole lobby in Europe. They just don't like wheat. Monsanto gave it up. I know they were using Genesis and other things else for hybridization. They went on. Simmet was a partner, and fizzled out gradually. So, but that does not mean people should not try to do it. Maybe they should pick up genes which are not readily available. By all means do it. Okay? And there are many things which the conventional routes that we use are extremely difficult. And transformants will give you an easy solution. You can cut, paste and slice and insert any way you want, really. The only thing with wheat, when I look at it, I have been a bit critical. Genetic transfers have to be in the right homologous group. So if wheat has got a group A, it's got 3 chromosomes, 1A, 1B and 1D. If you insert a gene in 1D and it should go into 1A, you cannot do it organized manner with any other methodology and classical cytogenetics. So you have to set your objectives in mind. And against the genomes, you got to look at it. Is it a diploid, like mutation breeding earlier on? Barley, a diploid, excellent. Single locus change and you see the inheritance. Wheat, 3 genomes, very tough. You cannot change that allele across 3 at the same time. So, yes, we should be looking at
Lauren here and then Roll.
So you said that, with goals of 2050 and solving or stopping malnutrition and hunger, how does transgenics conflict with that end goal? I mean, you have things like golden rice, which have been promising, but have not been widely accepted because of people's fear. That is going to stop us from reaching those goals by 2050. How do we overcome those? How do you satisfy the public's desire for knowing what they're eating, consuming?
I think it's a lot of education, lots of advertisement, lots of policy setting, and lots of making it very clear that this is not possible any other way. And you are compelled and proving that it's not harmful. I think it's a fear. It's a fear, and that carries on. The lobby of anti-things is very strong. Researchers have done their part. Okay. So it's tough for a researcher, unless the man is like Norman Borlaug, we can go and see the president right away. Okay. There are not many like Norman.
Yeah.
So you think it's out of the researchers' hands now? It's now.
Researchers have done their job fantastic. They can accomplish things all over. I mean, that talent is all around the world. How do you sell the stuff in the market is difficult.
Nurul?
Yeah, it's a lot of questions coming up about the transgene, so I'm going to take a couple of minutes probably. Yesterday I was reading an article, BBC Science.
Can you speak
Yes.
louder?
That BBC Science says we have a new salmon. It's already approved by the FDA. It has gene from salmon to salmon. It's huge, and it will be
Salmon fish. Salmon fish.
Yeah,
Oh,
yeah,
salmon.
approved.
Salmon fish.
USA, we eating about 75% this product of the food product contains GMO. So we are okay with that. And okay, let me ask you. I'm asking to pass a comment. You have been working 40 plus years in cytogenetics and also in the field. How do you see the cytogenetics is going to impact or help in our breeding program? Is it needed? We are in a smartphone era. So cytogenetics, how is it going to play? I want to hear your comment.
If it's used in an applied sense, Noor, I think it is a very strong tool. I showed you a couple of sequences. I didn't get lost on those 4 Bs with 1 quadrivalent. I didn't want to find out which is pairing with which, okay? Now, the pure cytogeneticists would like to know in that 4 what are the chromosomes associated. So I did not waste my time on that, okay? The same thing with the A and D pairing on that ABD triploid with the PH1C. I did the in situ hybridization and I found out it's A and D, opened the door up for molecular people. I said, now you get your D genome specific markers and you apply it and you detect the transfer, link it with the trait, and you're on your way. So that's applied cytogenetics. Pure cytogenetics, I think, is going to be very tough. You're not getting people in. Most of them are retired out.
Do we need that? Do you need to get new generation?
I think there should be a balance. It should not fade away like mutation breeding has. I know mutation breeding was a bandwagon. I was part of it. 1963 right up to the first 2 decades, everything was ionizing radiation, cobalt and X-ray and whatnot, and fast neutrons. When nothing came out, then that storm went away. Then we've come on now. But I would say keep a parallel, let everything go. You don't abandon a section of science just because it is not there. That's injustice.
Yeah, people can't live in time. So, by the way, before we pick Hong Bin's question, it is now past 1. If you need to go to class or if you need to be somewhere, you're welcome to leave, but we're going to continue for another 5 minutes picking questions, okay? Thank you so much. Some people need to get to classes, we'll just give them half a minute to be out of the room and then we can
Okay.
continue the question.
I know your name.
I sent 2-3 emails
I didn't get it.
them, and I know what was wrong.
you sent it the FS mail.
So that I wanted to.
All right, go ahead.
Okay, my name is Hong Bin. I work genomics. Actually, we work on some of the synthetic lines. Actually, not from not from cement, from USD and North Dakota. You know, where Fargo, they are the Chinese guy work on the synthetic. Whose name is Steve Xu?
Leonard Jopper and Steve Shu,
Steve Xu, yes.
combinations with...
Yes, here we work on the 19 synthetic lines. There's a hexaploid synthetic line between stolen weight and the
Coming
POCI.
next year?
And what we found, basically, we work on both genomes part and the transcriptome. which is the RNA. And what we found actually, we counted the chromosome. We have work on F2. That means after double chromosome doubling, and you get hexaploid. From triploid, you get hexaploid. And we check the genes. We work on genomic DNA and RNA. We check the genes. Almost all the cases, that's 1 + 1 = 1. I don't know what's happening. I don't know if you have any comment. That means you have two set of genes, one from the, let's say, durum weight. There's another side from Toshi, and you put it together, actually, it's never 1 + 1 = 2. All the time, we analyze 19 combinations. I don't know what's happened. I just want to see you have any explanation on this kind of phenomena.
I don't have a crisp explanation, but I can make a suggestion. Now, all those 19 synthetics, I think you got the batch from Leonard Joppa. The Durham was exactly the same, Langdon by different.
Yeah, we have Langdon, yes.
I wish to get your concept of 1 + 1 and you don't get another additive effect. You would look at the diversity of the Durhams. Try some of the synthetics that you have here just as a sample and see what you analyze, because there's a wide diversity of the Durham germplasm in the synthetics which Amir has got and Jackie Rudd has got. Okay, he's got about 51 Durhams on the female background, and I could specifically tell you which 4 or 5 Durhams to take. And
We have about, I believe, over 10 different genetic background for Durum weight and both, and Toshi.
I would say diversify the Durham parents across the A, B genomes, then you might see something different.
Thank you.
Well, thank you so much, Dr. Kazi. He's going to be here for the next hour sitting in this room, so if you have any questions,